All right, guys, welcome back to section number 4. I am so sorry that this is very delayed. I have just been extremely busy with a bunch of other stuff. But anyways, that's my little crash out. But let's get into lectures 7, 8, and 9. 7 was in carbohydrates, if I'm not mistaken. Hopefully they didn't change it. I reviewed the slides. Number 8 is glycolysis, and number 9 is Krebs cycle. And don't mind me, I'll be drinking coffee. And sorry for the noise. I'm recording this at Andrews right now during lunch hour. So let's do this. So for lecture 7, carbohydrates, specifically, we're talking about monosaccharides. So there are two main groups that we can categorize all monosaccharides. The first one is aldolases, which just means that we have an aldehyde, as demonstrated here on this diagram to the left. And the next one is ketoses, which it's a ketone, as shown by the name right here. And there are D and L. In previous lectures, remember that we said that amino acids are only on their left, or we only find L amino acids. Well, in this case, we find them only on D. So right here, it says all the sugars in our body are D sugars. So just remember that they're opposites to each other in terms of what's naturally found. And then in terms of what is D, what is L, it's the same thing with like the amine thing here, except we're talking about the OHs on the fifth carbon, the penultimate. And I'll explain why a penultimate is important. Not right now, but just know that it's the fifth carbon, not the first or the second, it's the fifth. And the sixth one, it's not either because it's not a chiral center. So yeah, that's everything on the slide in terms of that. So here are a bunch of aldolases, ketoses, all that, or aldolases, aldoses, and ketoses. And the ones that I highlighted in red are ones that you should spend some time memorizing in terms of which ones you have to prioritize, and not only just for this exam, but also for the MCAT as well. I remember they had us write a Fisher projection. The question was multiple choice. Out of these four Fisher projections, which one is the correct one? And so they would give four examples of this ribose right here, and we had to just pick the right one. And that was just rote memorization, unfortunately. So it was pretty hard to try to get it. I mean, they did give you a Haworth projection, so if you knew how to convert from Haworth to the Fisher, you were fine. But also having it memorized just made it really fast. So the ones that are highlighted in this red rectangles, I really recommend. They're classified by the number of carbons, and then also the aldoses or ketoses. So that's just on this slide. And definitely, like if you had to do a hierarchy, I would prioritize the 6 carbon aldoses and 6 carbon ketoses. Those are the ones that you should just know off the bat, especially glucose, mannose, galactose, fructose. Those are the number ones you guys should know. And then you can work on the 5 carbons afterwards. The 4 and the 3 aren't as common, but if you have to choose between either the 4 or the 3, I recommend the 3, just because more of the intermediates in glycolysis are these 3 carbons, not the 4. The 4 is only in these anaplerotic reactions and Krebs cycles, so don't even worry about that yet. Uh, so moving on to cyclic monosaccharides. So in aqueous solutions, linear sugars can form cyclic structures. What this just means, and this is exactly why I told you, focus on that anomeric carbon, the number 5, is or this special fifth carbon, is that when we're showing this mechanism, right? This is the Fisher projection at this is the like we're facing top down, and then we tilt it to the side, if that makes sense. And this is what we get here. We're just tilting it essentially. And in this picture, we see that this is the fifth carbon, and it's that hydroxyl group. So it's shown right here, and it's attacking our carbon number 1 where the aldehyde is, or if you're working with ketones, in the second carbon. So just know that subtle distinction. But the reason why we're interested in this is because for aldoses, we're going to form a pyranose ring, which just means that we're forming a 6 membered ring, one being an oxygen, obviously from the hydroxyl that combine. But we're going to see that this carbon number 1 is special in that it's an anomeric carbon. Now, what do we mean specifically? The best way and easiest way to find an anomeric carbon is to find the carbon that's connected to 2 oxygen atoms. Now, this carbon is the only one that is connected to 2. The rest of them are only connected to 1. If you just go through this, you'll see that all of them are connected to 1. They're not connected to 2. Carbon 5 is only connected to 1. It's this one right here. And the top one is just a carbon and then a hydrogen. So anomeric carbon, just easiest way to identify is to find the carbon that has 2 oxygen atoms connected to it. And then in terms of alpha and beta, the thing with the alpha carbon is it can be either C1 in aldoses or C2 in ketoses. And when you have long strings of them, it can be very convoluted to find the anomeric carbons for each monomeric unit. And so that's just something like a good rule to have in mind. But then additionally, there's alpha and beta. So with anomeric carbons, what ends up happening is you have this like almost equilibrium of this state and this state where there's a beta and alpha. And all this means is that we're looking at the hydroxyl that's attached or that would be formed when you have this anomeric carbon forming. So like the aldehyde right here, this carbonyl turns into a hydroxyl, right? Because of this intramolecular reaction. And so this is the alpha, just meaning that the hydroxyl is going to be pointed in the opposite direction of number 6, opposite orientation of the highest numbered carbon. So this is our carbon number 6 pointed upward. This one's pointed downward. Whereas beta, they're on the same side. So it's pointed upward. This one's pointed upward. And so what I was telling you about this equilibrium is this mutarotation, which is just the interconversion of alpha and beta forms. And then furanose are just 5 carbon rings, pyranose are 6 carbon rings. And then the Fisher to Haworth projection, basically the way I like to look at it is like this is a Fisher projection, right? And you have your substituents coming out from this way and also the other side, right? What if we just let it fall to the right? Then the stuff on the right are below the plane now, and then the stuff on the left are on top of the plane. So when you're doing these Fisher projections, you'll see that, for example, we'll talk about carbon 6. It's a very special case and in terms of the way it cyclizes and everything, so don't worry about that. But for example, number 2, we see that it's on the right, so it's going to be pointing downward, so it falls down. Number 3, it's upward. Number 4, it's downward because again, it's the left and right. And then number 5 is just forming that intermediate carbon. It doesn't form an up or a down. But yeah, we'll talk about number 6 in terms of meter rotation. It's a little bit finicky in terms of how it works. And then I think the slide is just supposed to demonstrate the mechanism. This is like the basic orgo one, where you just have an alcohol group attack an aldehyde, form a hemiacetal, and then from the hemiacetal, well, this is technically an hemiacetal, and you know that you could also, if you remove water, you can drive it to a full-on acetal, but that's not important in this case. We stop at the hemiacetal with cyclic monosaccharides. So moving on to the chair conformation, you'll be able to see alpha beta much more clearly with this. So beta D glucose is the most stable monosaccharide, where all of our substituents are equatorial. That's exactly why glucose is our main monosaccharide in all of our cells. That's why it's the energy molecule. It's because it's the most stable to be transported through the blood. It's not energetically unfavorable to keep it in that state. So when we're talking about the beta D glucose, it's this one right here, where all the substituents are equatorial here. They're not in the axial positions. And as I said about alpha beta, right here you can see the OH on carbon number 1 and relative to the carbon number 6, that they're in opposite orientations, right? This one is down, this one is up. Yeah, because on carbon number 5 right here, this is it's not going down in this case. That's the hydrogen, but we're not comparing it to that. If it were to be beta, then we see that this goes up along with this connection right here from 5 to 6. It also goes up as well. So that's something to keep in mind. Now, if we were to flip it, then you could say like this is down and this could also be down to form that beta as well. If we just rotate it 180 degrees. I don't want to confuse you, so if that didn't make sense, just ignore it. Then there's D and L. Now, D and L chemistry, you can really only like determine it if it's in the Fisher projection. That's realistically where you can detect it. And then furanose, pyranose, all that means is the number of carbons that, or sorry, the number of, yeah, the number of carbons in the ring. Sorry, it's what the pyranose means is you have a 6-membered ring, and then in 5 carbons, and then the furanose you have 4 carbons but with an extra molecule of oxygen, so that forms a 5-membered ring. So that's something to just know definitions wise. Here's some practice problems in terms of 5 carbon aldolases. Identify each epimer pair. You can just run through the slide deck. I would have posted it by now. So run it through. These are just some examples for you to go through it. But I didn't tell you about epimers. That's right. So sorry. Essentially, the best way I can tell you what epimers are is that if we look at, for example, glucose and mannose, right? They're very similar, as in, for example, they're similar at this carbon, carbon number 3, carbon number 4, carbon number 5, and then this is just the same, carbon 1 and 6. What's different is carbon number 2. They're alternating the OHs. Because it's different at one carbon, that means it's an epimer. An epimer, it could be at any specific carbon besides carbon 1, because it's an aldehyde in this case. But for carbons 2, 3, 4, 5, 6 is not applicable because it's not a chiral center, remember. This applies to chiral center carbons only. So carbons 2 through 5, those can be alternated, and if only one of them is changed, then it's a epimer. But now, if they're mere reflections of each other, then that is not an epimer, that is an enantiomer, because we know that they're reflective images of each other. So that's something to note as well. So that should give you what you need to solve this, at least. So my apologies for that. I should have included that in the slide deck. Maybe I can talk about it later. But monosaccharides are reducing sugars. What that just means is, as you can tell here, when it's cyclic, we have an alcohol. When it's Fisher, we have a aldehyde. And then when we when we oxidize it further, meaning that the sugar is a reducing agent, it will convert it from a aldehyde to a carboxylic acid. So if sugars inherently are reducing sugars, they reduce other things. So hemiacetals can easily reform aldehydes. So it goes from this to this. That's all that's saying. Linear aldehydes can be oxidized to acids, and they reduce other molecules as a result. Like remember, for every reaction there's an action, or whatever the saying is for Newton's laws. And then ketones may also react with oxidants, but much more slowly. So ketones are extremely stable just because they have the two carbonyls. The carbon is being stabilized by two different alkyl substituents, whereas aldehydes, they have a hydrogen right then and there, which makes it more polarized. So that's the reason why ketones, they may do it, but they also just they don't go to the full extent. And then there's also the other argument where they can't oxidize into a carboxylic acid because they have the two alkyl groups and you can't get rid of those. Um, and then glucose can be oxidized at the C1 or C6 position. So here, or the C1. So that's what these red arrows mean. And it could be either. So oxidative cleavage can form other molecules where you either form 5 carbon, 4 carbon. You're going to see these more in the Krebs cycle and also the glycolysis phases. So just something to keep in mind. But just a fun fact, the blood glucose is determined by the oxidation of glucose, which is this, to its lactone, a 6-member ring with the ketone. But I know this is gluconate. It would be showing you the lactone equivalent of it. So it's just a fun fact. Uh, Disaccharides. So we were just talking about single subunits, right? But we can combine them together through glycosidic bonds. This thing right here, between the anomeric carbon, right here. This is our anomeric because again, it's connected to oxygen to with another subunit. So this allows us to form two unit sugar. So essentially, the basis of a disaccharide, the way you do it mechanistically is you have an hemiacetal right here, and then you also have an alcohol combining together. So this is a very complicated structure. All I really want you to focus on is that you're having this anomeric carbon react with a hydroxyl group from a different sugar. So hemiacetal plus an alcohol, you form this disaccharide. And it's the same thing with also polysaccharides as well. That's how you keep adding them like Lego bricks, essentially. And then you have reducing sugars, which have that free anomeric carbon. In this case, we have this one right here, an alpha D glucose. And non-reducing sugars just means that there are no free anomeric carbons. So the example I can give in this case is, let's say we weren't just forming this disaccharide, right? Let's say we were trying to form a chain and we now have this 2 subunit and we're trying to add one more. So this is a non-reducing sugar, this alpha D glucopyranosyl. Don't worry about the names. Just this subunit right here is non-reducing because the anomeric carbon is not free. You don't have this free OH. It's participating in glycosidic bond. However, for the other one that was just attached to our 2 subunit, there is a free reducing sugar right here. There's this hemiacetal that has the OH that's free. So this one can participate in glycosidic bond. So that's the distinction between reducing and non-reducing. It's very, I don't think it's even possible to find a single monosaccharide that has a non-reducing sugar that isn't oxidized. So you can oxidize this to form that lactone as we were talking about, and you can look up the structure. The lactone cannot participate in forming a disaccharides or polysaccharides. So that's just in case, but you guys have probably gotten homework problems on it or whatnot, but that's just a distinction between reducing and non-reducing. And then all residues besides the last residue with the naming N with a sil, and the final residue ends with the side or os. And if reducing, this is like little nomenclature tips, is that if it's a reducing N, it's going to end with an OSE. So in this case, on this disaccharide to the right, we see that ends with an OSE. Whereas if it's non-reducing, it's going to say O side. So if let's say this this anomeric carbon was not free and it was a non-reducing sugar instead, this glucopyranose would not be glucopyranose, it would be glucopyranoside. So that's just the difference between the two in nomenclature. It'll help a little bit in terms of memorizing all the nomenclature. That's not the whole point. The point is that you're able to picture the molecule in your head, the properties, the chemical properties, whether it can participate in glycosidic bond formation, whether it's a disaccharide or a polysaccharide, alpha, beta, D, L. That's really what we're trying to get at. And then pyranose, and then if it's gluco and aldose or ketose, it's not about putting it all together and then, just naming a structure just by looking at the structure. That's not what we're trying to test you on. And then here's a practice as well. So name the type of bond that's shown here. Obviously, like It's, and I mean specifically, so here's some practice. Name that bond, and I want you to name specifically which carbons it's connected at, whether it's alpha, beta, glycosidic bond. That's mainly what I want you to get at. So the answer's there. And then also to find out whether it's reducing or not. Same thing here. Do the same thing. You can go through the slide deck for on. Moving on to polysaccharides now. So we're going to combine a bunch of those monomers to form our polysaccharide. So one case study I want you guys to really focus on is starch. And starch essentially is a storage molecule for plants, and it has a very rigid structure, meaning that it's very stable just because of those glycosidic bonds, the way it connects. So right here's an example of starch after forming these branches and everything. So with starch, it's a branch polysaccharide made of glucose monomers, and there are two motifs with it. There's amylose and amylopectin. Amylopectin. I don't know how you pronounce it, but so with amylose, there's an alpha 1,4 linkage. What that means is if we look at this diagram right here at this red rectangle, we see that we have our carbon 1 alpha and then our carbon 4. That's where it's being connected to this glycosidic bond. So this chain right here is a demonstration of amylose. Now, amylopectin allows this motif of, again, you're still having those long chains, but now you're introducing a alpha 1,6 linkage. So at certain points in these like long chains, you're going to see one, glucose unit from carbon 1 interact with the hydroxyl group on carbon number 6. And that's what allows you to now change directions and form these different branches. So this right here, these branches sticking off are alpha 1,6 bonds. So that's just like the difference between them. This kind of parent strand or parent chain is our alpha 1,4, and then where it breaks off is the alpha 1,6. Now, where it forms these smaller branches, the chains, and it goes outward, that's considered our alpha 1,4, right? And then there's another alpha 1,6 chain here for each of them that forms just like the 2,4, like it forms like a fork essentially. So something to keep in mind in terms of what their functionality is in terms of structure. And then there are storage advantages to using starch because it reduces the number of dissolved sugar residues in solution, therefore decreasing osmolarity. In simple terms, starch, because it's very polar, it'll basically like form this like aggregate, right? And because it forms this aggregate and tights very packly, we can reduce basically if we were to have a fixed space, right, of in a cell, and we're able to reduce the space that these starches can take up within the cell, we're able to add more sugars as a result. So it serves as a better storage rather than just have free glucose monomers just floating around in the cell. If we're able to pack them more tightly, we can fit more in, and that's the whole purpose of starch. And then there is one reducing end that reduces reactivity in terms of these starches. Like as you can tell here, on this diagram, you see that this arrow is saying that there are reducing ends, and this is where we're adding the glucose units. So it's almost like, again, like think of it like Legos. You're adding them one at a time, and the chain just increases as you go on and on. And then branching enzymes that harvest glucose from polymers work from non-reducing ends, and then simultaneous cleavage of glucose from non-reducing ends of branches. So that these are, I'm talking about specifically, if we're trying to get rid of these glucose units, we go from the opposite side, where we're getting rid of them from the non-reducing ends right here. And then what do glucose residues and starch hydrogen bond with to stabilize it? Water. That's the case. And so it allows for that better retention of water. And that could explain why, as like humans or plants, like we retain a lot. So for this second point, simultaneous cleavage of glucose from non-reducing ends of branches, what it means is that, again, we're working from the non-reducing end, the opposite side of where we're adding them, right? And we're going from the branches that are closest to that farther end. Does that make sense? So if we're looking at this diagram right here, right, we would cut off this branching right here. I'm looking at the purple line, not the blue. I would cut off this branch right here, but I wouldn't cut off this one first. It would be one at a time, almost like you're taking away one brick at a time. So you're adding one brick at a time, and you're also taking away one brick at a time. That's the best way I can conceptually think. So other polymers, polysaccharides that you guys should know, there are glucose polymers, so cellulose, glycogen. Just know their characteristic linkages. So for example, cellulose has beta 1,4 linkages, glycogen has alpha 1,4 branches with alpha 1,6 linkages. And the difference between glycogen and amylopectin is that glycogen has more branches than amylopectin. So if we go back to this previous slide, and I may not have mentioned, amylose and amylopectin are two different kinds of starches where they can combine together and almost have this like hydrogen bonding within each other, as demonstrated by this graph, but they are two distinct motifs essentially. And so in this case, we have glycogen, which again, it's very similar to amylopectin, but it just has more alpha 1,6 branching. So something to keep in mind as well. And then there's chitin, and which is basically beta 1,4 linkages of N-acetyl D-glucosamine modified glucoses. What that just means is it's this diagram on the left where again, it's a carbon 1 to carbon 4, and it's beta because it's up, and the carbon 6, our penultimate carbon, is up as well. So that's what it means. And then it forms a strong hydrogen, it's found in insects and fungi, and this is exactly what allows for the hard skeletal framework of. So peptidoglycans are essentially beta 1,4 linkages that are linked with D amino acids. Now recall, we said that our body usually contains L amino acids. So we have to modify those amino acids to go from L to D. And we have to do that through a chemical modification. We can't just like turn it around or whatever, because it's a chiral center. So we have to modify it first to an L amino acid, then attach it to a glycan, which is just a polysaccharide. So something unusual, but it forms a strong hydrogen bond network because, again, the amino acid is very polar. It has the amine group and the carboxyl group that can allow for that extra hydrogen bonding. And this is what actually forms the bacterial cell wall, and what allows it to either have, if you're doing staining, for example, not to get too off topic, there's gram-positive staining and gram-negative, and this just has to do with the prevalence of peptidoglycans in the cell wall. So something cool to note. So beta 1,4 are very strong because it allows for hydrogen bonding between glucose residues. Okay. So what's the difference between proteoglycans and glycoproteins? I was always confused when I first took this class, and then I think I parsed through it specifically. So proteoglycans, so you know how like in organic chemistry, not to bring trauma or scars or whatever, mental anguish, but essentially we have our substituents more upstream in the name, right? And that just means that they're less prevalent, they're less of like value, I guess, or less priority. Whereas if you have them at the end, they're more prevalent, like in OL, demonstrating there's an alcohol. So keep that same idea, but for this. So proteoglycans, proteo just means that we have less amounts of proteins in the glycan, in the polysaccharide. So it's typically, and again, some background in terms of proteoglycans, they're typically found near connective tissue. And this is just because we want to allow for some sort of connectivity to our polysaccharides, to our energy. So then now, on the other hand, there's glycoproteins, which is essentially, again, glyco now is the less, and then proteins is the more abundant. So there are more proteins than carbohydrates, and it tends to, you can think of it as proteins that are glycosylated in terms of our like biological context, if you were to bring this into real life instead of all theory. And so it allows for a large amount of complexity. It allows for unique fingerprints for protein recognition, cell to cell recognition, and it's what allows for transduction, external cellular matrix signaling into the cell itself. It's through these glycosylated proteins. In a little review, what amino acids can be glycosylated? O-linked, which is serine and threonine, and then N-linked at the asparagine residues. And then in terms of a little preview of bioenergetics, delta G is not the same as delta G not whatever. Basically, these are two different. And remember, overall though, delta G will tell you the energetic and the energetics, there's the free energy, that's what I meant to say. Overall, the free energy will determine if a reaction is going to go forward. But these two take into account two different conditions, right? For this one, the delta G naught, this one takes into account standard conditions where we assume that the cell concentrations are fixed, right? Or these concentrations of products and reactants are fixed. However, in cellular concentrations, we just know that's not true. We know that these are going to vary. The C, the D, A, B, whatever it is, that ratio is going to vary based on cellular concentrations. So this can actually alter whether or not the delta G itself is actually positive or negative, even though the negative delta G naught may tell you it's negative. In cellular conditions, it could turn into positive because this value is great enough because of that ratio. So how can we make an unfavorable reaction go forward in terms of biochemistry, in terms of just real life? We can constantly remove the product. So if we're forming a combustion reaction, right? No, it's not a good one. If Okay, so you guys are going to learn this concept in a little bit about pyrophosphate. All it is two phosphate groups being forced together. They have a huge negative charge and they want to repel outwards, right? Well, you can use this to your advantage because they want to split up, right? And if a reaction forms a pyrophosphate in the products, but you get rid of it immediately by another side reaction, well, then your equilibrium, you're going to drive it towards the products because of that. So that's one example where you constantly remove products. And there's another one called coupling reactions where the standard free energies, just like the enthalpies in Hess's law, again, this is connecting different concepts together, but it makes sense conceptually, is that standard free energies, you can add them together. So in this diagram, when we're trying to take this reaction from A to C, we can add these delta G values, and they're additive. And then coupling can create a net exergonic reaction. That's exactly why we have ATP for some of these reactions in glycolysis and Krebs cycle and ETC, photosynthesis, pentose phosphate pathway. We have ATP there to power energetically unfavorable reactions that are on their own. Reactions that on their own, they're energetically unfavorable. And so just before we get into glycolysis, I just want to talk about nucleotides and nucleosides. So nucleosides are only considering the sugar and the base. So the base is this one up here. And in this example, it's adenine. And then we're talking about the sugar. Now, that's a nucleoside, right? But a nucleotide is when we add all the phosphates into it. Now, a nucleotide could be a 1 phosphate, it could be a 2 or a 3. That's where we add this M right here, or M for mono, D for di, T for tri. And so these are again, nucleotides are building blocks of DNA, and they're synthesized by a different pathway, which we'll go more in depth in unit 4. And usually the sequence is nucleoside to nucleotide, then to being added to RNA or DNA. And then I was kind of foreshadowing this before, but ATP, its hydrolysis is extremely exergonic, meaning that it's very favorable, it's very spontaneous. And why? As I mentioned, when we hydrolyze ATP, you relieve that electrostatic repulsion. If you can see right here, we have so many negative charges that are so near each other that if we get rid of one, it's going to alleviate some of that negative repulsion. So again, that's you relieve that repulsion, you let it distance itself, and that's actually going to drive it forward. But also, the inorganic phosphates that are released, that are cleaved from the ATP, they're stabilized by resonance, and that's shown in this part right here, number 2. And then also, mass action favors hydrolysis. What this just means is water is a substrate in reaction, and a high concentration of water will push the reaction towards the products. It won't push it towards the reactants. It'll make it move the direction it needs to. That is ATP hydrolysis. And then there are also high levels of ATP compared to ADP and AMP in the cells. And this is exactly why, again, with the reactants driving to products, why delta G for the reaction is even more negative, is because in delta G naught, the standard conditions, we're assuming they're equal or there's something special that keeps them equal. It's basically not what's found in the cell. But in the cell, we know that ATP is at a much higher concentration. And so it just drives the reaction towards the products. So something to consider as well. Okay. I reviewed my slides ahead of time just because I felt like lecture 7 wasn't as good as that. I felt like lecture 7 wasn't well presented. But it's a hard lesson to talk about just because it's very much applications based, and I think just going through problems coming to office hours is really going to help out a lot for those. But it doesn't make up the biggest portion of the second exam. It's going to be more so all these pathways afterwards. Okay, but moving on to lecture 8, glycolysis and bioenergetics. So we're just going to cover the bioenergetics very first and brief. And so first thing that when we talk about bioenergetics, we're talking about these redox reactions where they're chemical reactions where our electrons are transferred. And so just to keep you orientated, I like to use an acronym called OIL-RIG, where oil stands for oxidation is losing, and then electrons is omitted from the acronym. And then RIG, reduction, is gaining electrons, omitted as well for this thing. And then two important terms to not get confused with the oxidation reduction thing is that an electron donor is your reducing agent and your electron acceptor is your oxidizing agent. So in this case, this table right here demonstrates that we have standard reduction potentials of some biologically important half reactions. So high reduction potentials just means that there's higher affinity for electrons. So on this table right here, we see that this has a 0.816. So the affinity for electrons right here is much greater than the electron potential right here for fumarate converting into succinate because it's 0.031. And notice how they're all positive. You may get some tables where they're negative like this, right? This is showing a half reaction where it's negative. Well, this one actually, this reaction, if you were to flip it over, forms a positive reduction potential, which just means that it likes to be pushed, spontaneously speaking, it likes to be pushed towards these reactants right here, the NAD plus, the H plus, the electrons. That's what the negative just means here. Remember, our delta G is the inverse of our delta E. In simple terms, all that means is that if the E is positive, our delta G is negative, which means our reaction is spontaneous. On the flip side, if E is negative, like this bottom one right here, then our delta G becomes positive because a negative times a negative is a positive. So that means that the reaction is non-spontaneous. So in this bottom one, we see that this reaction is not spontaneous in this direction. Now, we were to flip it where it goes from NADH to the reactants and dissociated products. Well, that turns into a positive plus 3,2O, and that means that it's a spontaneous reaction. And then for any redox reactions, when you're calculating this delta E naught, again, naught is very important for this. Um, you're going to take the reduced EI, whatever it is, minus the E naught that is oxidized. So when you couple reactions, couple half reactions, you're going to basically take the reduced half reaction minus the E potential for the oxidizing half reaction, and we subtract because the equation only uses reduction potentials. The minus sign takes into account that you have to reverse the sign for oxidation. So we would just leave it as this, essentially, this reduction. Now, if we were to flip it to 0.32 and it was coupled to some other reaction here, well, then it would be that reaction that we have, and we flipped it so that way, because this is a molecule that's going to become oxidized, and it'll be plus 0.32, and there wouldn't be a negative sign here anymore. That's just like, if that flew over your head, it's okay. Just keep it in simple terms like this, and go through practice problems, really trying to understand which is the molecule that's being reduced, which is the molecule that's being oxidized, and then from there you'll be able to determine that whole thing. And then also something to know is that when you're using this equation, delta G is equal to negative NFE. The N is equal to the number of moles of electrons transferred, not the moles of your molecule or whatever you're working with. And then F is Faraday's constant, which is 96.5 kilojoules per volt times mole. So something to keep in mind as well when you're calculating this. You should also, for the MCAT sake, to have this number memorized, 96,500 joules, or 96.5 kilojoules. So moving on to the next thing, there's oxidation states. This is essentially just counting the number of owned electrons per carbon atom. So in this case, for methane, we know that the dipole direction, or where the electrons are situated, are closer to carbon because the carbon has a higher electronegativity than hydrogen. So it's going to own technically all the dots that forms this covalent bond. So it's going to 2, 4, 6, 8. So that forms 8 total. Now, half the number of electrons in bonds to the same atom. So for example, in this case, ethane, for the C to C bond, instead of saying that these 2 electrons are to this single carbon, it's just shared equally because they're the same atom, they have the same electronegativity. So for each carbon atom here, each of them owns 7 electrons, and that's the oxidation state, I'm pretty sure. And then there are no electrons in bonds to more electronegative atoms. What that means is, for example, we're taking formaldehyde right here, right? And we're saying that the carbon only owns 4, and that's the ones that are to the hydrogen. It doesn't take any of the electrons from oxygen because more electronegative than carbon. And electronegativity, if you ever need like a quick rule, is this table right here, where hydrogen is the least, oxygen is the most, and then it sorts itself in between. And then here's a practice problem, like where you indicate the number of owned electrons for each labeled C. Just a word of advice is, for previous exams, mine that I took and also one that I graded for last year, they did have a problem like this. So be very comfortable in terms of counting the number of owned electrons per carbon that you're looking at. And so the answers are in the slide notes, so you can just check them out. Then moving on to electron carriers. And electron carriers, it's very much this, if I go back to this table, this NADH, the FAD, that's what those electron carriers are. And they usually transfer electrons in pairs. And so we have oxidation, which usually involves creating a double bond via dehydration. And so, for example, we have pyridine nucleotide coenzymes, which is just NAD plus, NADPH. And the oxidized forms are where they have the pluses in the names. And then the reduced are H's. Remember, just like a general Gen Chem principle, is that when we have stuff that's reduced, or even just orgo, sorry, a general Gen Chem principle and orgo, as well, as you guys have seen, when we're reducing, we're adding H's. When we're oxidizing, we're adding O's, or we're removing those H's. So that's just something to like keep in mind, to know kind of quickly what's the reduced form and what's the oxidized form. And then for these coenzymes, they usually carry 2 electrons and are mobile, meaning they move around the cell carrying these 2 electrons. And then there's also these flavin nucleotides, which are FAD and FMN. And the oxidized forms are just without the H's, and then the reduced are with the H's added to them, and usually it's 2. And same thing here, it's 2, but we just don't designate it. We added like a plus H proton here, so just know that this is 2 electrons still, even though it shows just the 1 H, whereas here, it's almost like it correlates to the 2 electrons that we're adding, or that's able to transfer. And then it can carry 1 semiquinone or 2 electrons, and it's usually bound to an enzyme. It's not freely floating like NADP plus or NADP or NAD plus. And there's an easy way to spotting redox reactions. Usually it's either the involvement of NAD plus or FAD. Usually it's changing the number of bonds to oxygen. So, no, it's my this my or thumbs up. Or number two, it's changing the number of bonds to oxygen. So for in this case, alcohol, there's only two bonds. Now there's or sorry, from the carbon, there's only one bond, there's two bonds, and then now there's three bonds to oxygen for that central carbon. And then three is the addition or loss of hydrogen. So those are like three ways to easily spot them. It gets trickier sometimes. The best way is to find the oxidation states. If they stay the same across a chemical reaction from reactants to products, it's not a redox. Now, if they change, it is a redox. Okay. And then now moving on to the glycolysis, the stuff where I feel a little bit more confident in is the cellular respiration. And I just want to give a brief overview before we get into the details, because I feel as though when you're first learning this, you feel like you have to learn every little trick, every little enzyme, cofactors, substrates. Let's just break it down into the bigger picture, and then we can worry about the little details as we go along through this review section. So number one is glycolysis, and all we're doing is just splitting the sugars, the glucose essentially, and we're giving partial oxidation of products. This is where we see substrate level phosphorylation. That's a key term to know. Substrate level phosphorylation just means that we're able to produce ATPs from our substrate. And that's different than oxidative phosphorylation, which we see in ETC. Oxidative phosphorylation just means, and I'll show this in slides, so don't worry, we will go over this. But oxidative phosphorylation simply just means that we use the proton gradient to drive our production of ATP. That's essentially what that means. Is oxidative, meaning that it's a redox reaction using protons, using electron carriers. We form ATP through that. Whereas substrate level is like, for example, we have the fructose 1,6 bisphosphate. It has two phosphates on it, and we just remove each one and put it onto an ATP. That's substrate level. So that's the difference between the two. And I just really wanted to mention that first. And then the second process is called the citric acid cycle, and it's the complete oxidation of the products of glycolysis to CO2. We break down our complex monosaccharide into carbon dioxide, and that's done by Krebs cycle at the end of it. It's both glycolysis and Krebs cycle combined to form that CO2. And then the last step is in order to oxidize the sugar that we have all the way to CO, we need to have oxidative agents, NAD plus, FAD plus, to be able to take the electrons from glucose and to be able to drive electron transport chains. So that's exactly what this third part is. This third element is now that we're done with the sugar and we extracted all the energy, now we need to put that energy into use and so that the cell can use, and that's through production of ATP from these electron carriers. And we'll go into that as like the third and final step. So there's two steps that involve our sugars. It's these first two. And the last one just has to do with all the energy that we've extracted from that glucose molecule. So something to keep in mind. And then important thing to note is that for the processes two and three of this, the TCA cycle and the electron transport chain, those are in the mitochondria, whereas glycolysis is outside. And this is important to know. Location is essential for you to know for the exam because a lot of practice questions can be derived from this, and also for future pathways too. So just a little tangent, but just know the locations. So, what is glycolysis? Note that glucose has so much energy inside of it, but we can't just release it on one step, or else our cells would literally turn on fire. And then, so the solution, in order to not set our cells on fire, is to have a stepwise breakdown of glucose, where we harness energy at each step, some manageable chunks, and turn it into ATP or reduce cofactors. So, as you can see, the net equation is glucose for glycolysis, not for the whole cellular respiration. For glycolysis, we're converting one glucose molecule into two pyruvates. So right here's the two pyruvates. Each pyruvate is 3 carbons long. So if we add 2 together, that forms our 6. And then at the end of it, the net, again, this is the thing, it's net. We form 2 ATPs that the cell can use and 2 NADHs. The waters and protons aren't really that important for this. I just really want to emphasize the ATPs and NADHs. And we can break down glycolysis into 2 steps. There's the preparatory phase and then the payoff phase. So I will go into that in depth, but just know that glycolysis, you can split it into 2 halves essentially. One where we put energy in, and then one where we take more energy out, essentially, from breaking it down. So again, glycolysis is just the stepwise oxidation of glucose where it occurs in the cytoplasm and it creates energy, ATP, NADH. And we're able to use that for one, the immediate ATP that we can use for other things in the cell in the cytoplasm, but the NADH, which can be transported into the mitochondria and be able to drive ETC, oxidative phosphorylation. And again, glycolysis is anaerobic, meaning that we don't use any oxygen. It is not present here. We don't need it. So this will occur whether we can breathe or whether we cannot. And again, there's two phases. There's preparatory and there's payout. And just like a terminology question for you guys to like ponder, the answers are in the notes below. But is glycolysis anabolic or catabolic? And it could get confusing, but short answer is it would be catabolic because we're breaking down glucose at the end of it. That's our definition of glycolysis. We're breaking down glucose into pyruvate, so it's catabolic, not necessarily anabolic, even though we have these ATPs that we're adding to it. So moving into the first half of glycolysis, there is the preparatory phase where it's steps 1 through 5. We're basically saying, okay, we have a glucose, but we can't really do much to it right now because in its base form right here. What we have to do is add phosphates to it. So that way we can now have energetics break it apart. And so that's what we're doing in this first half. So what I'm going to be doing for this, and just to preview it, I'm going to give you like a TLDR slide where it's just easy review. You can just kind of test yourself on it. And then I'll also give like an in-depth slide in terms of the kinase, all that stuff. So I'll just go through it very quickly with substrates, reactions, and all that. So just bear with me. So the first step is basically we have our free glucose right here. Our glucose is not phosphorylated. It's just base form. What we need to do is phosphorylate it. And I'll explain the next slide why we even do this in the first place. But we have to use ATP in order to achieve this. It just does not grab a random phosphate from the cytoplasm. It has to use it from ATP. So glucose plus ATP forms our glucose 6 phosphate. I want to emphasize 6 because the reason why we use that is because it's on carbon 6 where the phosphorylation occurs. So that's the reason why we say glucose 6 phosphate. Now, if we were to just make up a random molecule saying like glucose 2 phosphate, right? Well, then the 2 phosphate would mean that the phosphorylation is on carbon number 2. So just something to keep in mind in terms of why we name it this. And the enzyme that performs this is called hexokinase. And hexo meaning for the 6-membered ring, and then kinase for phosphorylation. It's a protein that phosphorylates things. And again, we're attaching a negative charge to the glucose, and this is the function of it, is that when we attach this negative charge phosphate groups onto, or negative phosphate group onto glucose, we're going to trap it in the cell because in order for glucose to go through the insulin-like receptor, or the glucose transport or whatever, it has to be that base glucose, this thing right here. So this will travel freely in and out of the cell. This will not. It's going to stay stuck. It will not be able to pass through that channel because of this negative charge. So in order for the cell to keep like its own storage of glucose, it has to add this phosphorylation to it. So keep in mind for like the exam, if the reason why we have this first step is because we want to trap glucose, but also because we need to prime it for the payoff phase as well. Okay, so moving into like the more in-depth is right here, this part. So we have this is a negative delta G. Now, you may think, well, we're adding a phosphate. Doesn't that mean that it would be almost anabolic per se? Well, no, because we're coupling reactions. Remember, when we couple this hydrolysis of ATP, we know it's highly exergonic from that previous lecture. So because it is highly exergonic, when we add this delta G plus this one, which should be positive, we're going to get a net negative. And so this negative delta G becomes negative 16.7. You don't need to memorize the negative 16.7. Just know that this first step is a negative delta G because it has that reaction coupling in it. And then for regulation's sake, because you should know regulation, glucose 6 phosphate acts as its own inhibitor on hexokinase. And the reason why you want to regulate this step is because we don't want to overuse our ATP within the cell. If this occurred at its own rate without any regulation, there could be instances where we want to produce glucose because we're fasting or whatever. And we want to produce this in our circulation. So we don't just want to always go forward. We may want to say, hey, put the brakes. And that's exactly what glucose 6 phosphate will do, is it'll negatively inhibit hexokinase and decrease this rate. So that way we can push the reaction rate towards the other direction with a different enzyme. So something to keep in mind. And then for step 2, TLDR, basically we have our glucose 6 phosphate. Now we need to convert it into a furanose ring, a 5-membered ring, fructose 6 phosphate. So we're not doing anything chemically. We're not adding bonds. We're not adding anything. All we're doing is we're changing the ring size from 6 to 5. And what does that enzyme usually mean, or which enzyme is it? It's a phosphohexose isomerase. Now, to break this word down, phosphohexose means that it's a phosphate group attached to a 6-membered ring. And then isomerase just means that we're this ring into a 5. we know we're not saying that it's specifically changing to a 5. It could have been a 4 or something like that, but we know it changes it to a 5 over the rest of the rings because it is most stable. It's more stable than those other alterations. A 4, we know that a 5 ring is more stable than a 4 ring. We know that a 5 ring is more stable than a 3, and so on and so forth. So just something to keep in mind. And then it makes structure symmetrical and exposes C1 right here for phosphorylation in the next step. That's the whole purpose of this step, is that we're going to phosphorylate this again, but in order to do so, we have to basically form carbon 1 that sticks up, essentially, or sticks out instead of being in the ring itself. So then here's a more detailed breakdown. Again, this one is a endergonic reaction, which just means the delta G is positive, but it's very close. The concentration of products and reactants are going to be almost equal in ratio wise, because if we know delta G was 0, then our equilibrium constant means that it's 1. And it's just from the equations. But this just means that we're going to have similar concentrations of fructose 6 phosphate and glucose 6 phosphate if this reaction were just isolated and continued for an infinite amount of time. And again, this can be reversible, but it proceeds forward through quick removal of product as it's used up, right? So this fructose 6 phosphate is going to be immediately turned into fructose 1,6 bisphosphate in the next step. And because we're converting it immediately, this allows us to drive the reaction forward. So moving on to step 3 is we're essentially adding a second phosphate group. So again, we just formed this fructose 6 phosphate. It's a 5-membered ring now. We prime that first carbon. So now we're going to phosphorylate it again using a coupled reaction using that ATP. And now we're using an enzyme called phosphofructokinase 1. And PFK1 is very important to know. Breaking down the term, phosphofructo. Phosphofructo just means that we have a fructose 6 phosphate. So the phospho is just the phosphate group that's attached to our fructose. And then the kinase just means we're going to phosphorylate it. So this tells you the structure, the substrate, and also the function of it. So that's what phosphofructokinase does. It attaches a phosphate group from ATP into our substrate, and it forms this fructose 1,6 bisphosphate. And so this is our commitment step. If we form this product, it will become our pyruvate and we will continue glycolysis. This is like the last energy investment, and then it's just like a roller coaster down from here. So just know that this is a very energetic molecule right here as well. So moving on to more of the specifics. This is the exergonic reaction because of that ATP hydrolysis coupling. And then it's the second step in this first half of glycolysis. We've been talking only about one half where we have to use ATP again. But this is just the preparatory phase. And one thing to note about this enzyme is that there is allosteric regulation. Okay, yeah. It's activated by AMP, ADP, and fructose 2,6 bisphosphate. It is important, the regulation of it, but we'll talk about the specifics of fructose 2,6 bisphosphate in a different lecture where it's like gluconeogenesis, insulin. We'll talk about that more in depth, but for right now, just memorize this. When we have these 3 molecules present, it's going to stimulate the forward direction. It's going to stimulate this enzyme and positively allosterically regulate the direction of it. And then it's inhibited by ATP and citrate. The reason why it's inhibited by ATP citrate is that these are molecules signaling high energy. If we have a buildup of citrate, that means Krebs cycle is being backed up because we have so much energy. We don't have enough cofactors to keep the reaction moving. And ATP, as you can tell, is like, if we have a lot of energy, we don't need to create more. Again, this is our commitment step. So if we need more energy, we want to commit to this step. But if we don't need that energy, we want to halt it. So that's why ATP would normally inhibit. Now, the activation by AMP means that this is a low energy molecule, meaning that if we have a high concentration of this, we want to create more energy because we're low energy. And same with the ADP as well. The fructose 2,6 bisphosphate, I wish I can give you an explanation right now. 1, I don't remember, but 2, it's just a memorization thing. And then question, why would you want to regulate this step when the first step is already regulated? And that's because our first step is not glycolysis specific, meaning that the first step could be for the pentose phosphate pathway, making glycogen. And this step is our committed step for glycolysis. So we want to heavily regulate this one very much. And then this is just a graph demonstrating the Michaelis-Menten curve or whatever, just a curve of enzyme activity and substrate concentration. So if we have high ATP, we see the enzyme activity is delayed by a lot. It's negatively regulated with ATP, whereas low ATP, it stimulates this activity to increase a lot more faster at a lower fructose 6 phosphate substrate concentration. Okay, so step 4. So what ends up happening is for step 4, we now have this high energy molecule. We've already committed to glycolysis. Now we have to follow through. What do? Because of this high energy and these two negative electron repulsions, we're going to split this molecule in half using aldolase. It's going to cleave it, and we're going to form two 3-carbon products right here. So aldolase is the one that does this, and we basically form two of these smaller carbon molecules. Each of them have a phosphate respectively. So this one, dihydroxyacetone phosphate, has a phosphate. This glyceraldehyde 3-phosphate also has a phosphate as well. So instead of one molecule having 2 phosphates, we now have one molecule and one molecule each having their own respective one. So they're still highly charged because of that negative phosphate. It's just now it's equally. Okay, so we see that this delta G is a very much positive delta G. It's 23.8 kilojoules per mole. So how could it be spontaneous, or how could it move forward at all? Why wouldn't it just want to stay in the substrates? It is true. However, these two molecules are going to be immediately used up in step 5 right away, or step 6 to proceed through glycolysis. So that's what it's going to be used. So one of the things to note is that if we're removing this constantly, well, then it's going to push our reaction to the right because we're assuming that this is negligible concentrations. So that's the whole reason why, despite it having a high delta G, we still see the reaction go more towards the forward products. And just a little like nomenclature stuff, dihydroxyacetone phosphate. So dihydroxy just means that you're supposed to have 2 hydroxyl groups attached to your acetone backbone. So we know acetone is our ketone that's attached to a methyl and a methyl. But we know in this case, each methyl group is going to have their own respective hydroxyl. Now, I know that this one only has 1 and then the other one is a phosphate. Well, that's the reason why we say phosphate, is because it's supposed to have 2, but we end up taking 1 away by making a phosphorylation. And then for glyceraldehyde 3-phosphate, what we have here is glyceraldehyde, which is essentially just, or it's like our acetone, except instead of it being a ketose, it's an aldose, meaning that we have an aldehyde here. Instead of a ketone, we now have an aldehyde. But what's it called? So glyceraldehyde, again, a 3-carbon molecule, and we have an aldehyde rather than a ketone. And we see that we say glyceraldehyde 3-phosphate because this is carbon. I know it says 4, 5, 6, but that's just counting the carbons on our substrate. So in this case, we have carbon 1 on the aldehyde, carbon 2 on the middle one, and then carbon 3. And we're seeing that the third one has that phosphate group. And number 2 has the hydroxyl because we know it's a sugar. I don't want to go back all the way to those slides, but there was one where it talks about in the sugars where glyceraldehyde and whatnot. So that's mainly what you need to know in terms of nomenclature, delta G. Yeah, again, this is the energonic reaction. This is what we're saying is delta G is not the same as delta G naught because we're taking into account our concentrations of products divided by concentration of reactants. And if we have no concentration of reactant or products, this is going to drive this to become much more negative and to push the reaction forward. So something to keep in mind. So this is the aldolase mechanism. I just recommend that you guys know this in terms of arrow pushing. It could be something very similar to what was shown in your first exam. So this is just go through it. Basically, we have the important residue lysine and aldolase, which acts as our nucleophile. It's going to attack the carbonyl of our fructose 2,6-bisphosphate. This is what that molecule is. It's just a Fischer projection, and it attacks it, and you're going to form this tetrahedral intermediate, right? And then our other basic residue is going to deprotonate our lysine amine, and it's going to basically collapse the tetrahedral structure, right? And from there, you're going to have a free, I think it's a water molecule. Yeah. You're going to have the carbonyl that you originally had is going to basically get taken from the tetrahedral intermediate when it collapses. So it's going to form that free water. And so now you lost that carbonyl, but now it's attached and forming this protonated Schiff base, which is just essentially the C and double bond. And so now with this, you have another basic residue, a different one. You don't need to know the specific amino acid, but it's going to deprotonate carbon number 4, the hydroxyl on carbon number 4, and you're going to form again you're going to form this cleavage of your fructose 2,6-bisphosphate. And so from there, you're going to form your glyceraldehyde 3-phosphate product first. And then from there, what you have to do is now we have to get rid of our Schiff base that we originally had. And how we're going to do it is remember that first base that deprotonated our lysine amine. Well, it's still going to be protonated, and that's actually what's going to allow for this last substrate or product, whatever, to dissociate from the enzymes. So electron lone pairs on this nitrogen that are going to basically form this double bond between the carbons, and that carbon-carbon double bond is going to deprotonate that base. And so now we have this, right? Well, if we essentially introduce water, it's going to go through this mechanism again and deprotonate this thing right here. So you don't really need to know the very end of it in terms of how Schiff bases are hydrolyzed, but just know that we're adding water at the very final step to release our second to last product. So this is just a brief overview. This I would put on like the last things to know for this exam. Like don't make this the center, but definitely you should know this mechanism. Step five. So this is the last step of the preparatory half of glycolysis. So we have triose phosphate isomerase. So what ends up happening is we form our two products, right? They're distinct molecules, but what we're going to do is we're going to form one molecule that's favored throughout this glycolysis pathway. And so we're going to create two copies of useful molecules. So what this means is our dihydroxyacetone phosphate, it can't be used in the rest of glycolysis. We have to convert it to glyceraldehyde 3 phosphate. So when we go back to step 4, right here, we form both of these in equal concentrations, right? Well, we don't want this because it's not useful in glycolysis. So we have to convert it into the other form through this triose phosphate isomerase. And I just want to point out that enzyme name is triose phosphate, meaning that it's a 3-carbon sugar. So 1, 2, 3 phosphate, which has a phosphate on it, and isomerase. And this is the same too. Again, it's reversible. But I just wanted to point out that a lot of these enzymes, they will describe what their reactants are, or substrates, or even products. Sometimes they're flipped, but just most of the time, this is a good rule to follow. And then just more in detail of step 5 isomerization, it's basically an endergonic reaction at standard conditions. However, it will proceed forward if we remove this glyceraldehyde 3 phosphate. Again, we already committed to glycolysis. We're just going to go straight through it now. So this concentration of glyceraldehyde 3 phosphate is going to go down immediately. So what would happen, and this is a question for you guys to just ponder, what would happen if there was a mutation in triose phosphate isomerase that decreased its catalytic efficiency? And you have to think, okay, well, if this step is slow, what ends up happening? Well, just think of it as like a bottleneck, right? Glycolysis would have a hard time proceeding, despite it being after the payoff phase. You would see this bottleneck where you're basically not necessarily going half the rate, if you know what I'm trying to say, but you're forming some of this dihydroxyacetone phosphate, which isn't immediately converted to this useful molecule. So you're going to have a buildup of this, and it's going to basically like bottleneck. And so that's what this explains. So now we're moving on to the second half of the payoff phase, step 6. So I just want to note something right now is that every step from now on is doubled to account for the 2 3-carbon molecules formed during prep phase. So for the step 5, we now have 2 molecules of glyceraldehyde 3 phosphate, and that's what we're going to be using. So everything is doubled in this case when we're counting it. So in this case, like I know it says 2 NADHs, this has already been doubled. So that's the 2. We formed the 2 ATPs as well. We formed the extra 2 ATPs here. It's technically, if you were to divide it in 2, this would be then accounting for 1, but that's what the parentheses are here for. It's to say that there are 2 of these molecules. Therefore, you need to multiply everything by 2 here. So just wanted to keep those straight, essentially. So now we're talking about just one molecule at a time. For step 6, TLDR, what we're doing is we're taking this glyceraldehyde 3 phosphate and we're adding a phosphate group to it, right? And we're forming this 1,3-bisphosphoglycerate. Now, what does that mean? You may think, Alex, well, aren't you phosphorylating it? Doesn't that mean you need to add energy to it? Not necessarily. Now I'll explain why. Remember how we talked about redox reactions at the very beginning? This is where it comes into play. And remember, we talked about NAD plus, NADH being an indicator that it's a redox reaction. So as you can tell right here, this is a aldehyde, right? And this product, I know it's not apparent at first, but this is a carboxylic acid right here, essentially. That's the way I like to think of it. It's a carboxylate group. You know how we lose this H, this CH bond? There's energy in there, a lot of energy, and we're breaking that bond and adding the O. And where does that energy go? Because remember, the carbon owned those 2 atoms, right? And now it no longer owns them. So where did those 2 electrons go? They went to NADH. That was able to power it. And so, yes, it's being technically phosphorylated. That doesn't mean that it was from ATP. It's just a random inorganic phosphate from beforehand. So in terms of, this is not a paying in step per se. You're not adding ATP. You're not coupling it. You're just simply oxidizing your sugar and you're reducing your NAD plus into NADH. So that's the whole reason why this doesn't have a high positive delta G. It's because we're undergoing this oxidation part. So yeah. So moving into the details of it, why? So our first energy producing step in glycolysis where we form our NADH, and actually, yeah. So we use this enzyme called glyceraldehyde 3 phosphate dehydrogenase. Dehydrogenase should really only be used for oxidation reduction reactions. And so whenever you see dehydrogenase, think NAD plus, NADH, FAD2 plus, or whatever, FAD plus, and then FAD2H2. Think of that whenever you hear the word dehydrogenase in an enzyme. And again, we're transferring the H from the substrate to our coenzyme, right? And electrons are removed from G3P and transferred onto NAD plus. And this step is endergonic and reversible, but it is driven forward since step 7 is highly, highly exergonic. So it's almost like we're removing the products right away. And no, the reaction occurs twice because there are two molecules of G3P. There are two of these, so we're now forming two of these coenzymes first. I know I keep saying cofactors. If I ever say cofactors for NADH or NAD plus, I really mean coenzymes. I just butcher it sometimes. So step 6. So this is the redox step where we're calculating essentially the number of electrons that are owned and the number of electrons that are resulting from the sugar and that. So you can walk through this again. For step 6, we have 3 electrons that carbon owns, 1, 2, and then this is 3 because it's a equal sharing. It doesn't own any from oxygen. And then 4 here and then 5, but these are not changing. So these are the same. And then here it's 1 now because it's only from this carbon. The oxygens now own these two, right? So where did the 2 electrons that were from here that were lost in the products? Well, that went to NADH or NAD plus. It's a form NADH. And NAD plus has 2, will carry those 2 electrons essentially. So this is step 6 mechanism where we're converting G3P into 1,3-bisphosphoglycerate. And this is, you guys don't really need to know this super, super in depth, but it is good to know because it could be one of those like little details that they test you on. So recommend going over it. Basically, I'll just go through it really quickly. You can skip the section if you want. So we have a glyceraldehyde 3-phosphate, and we have a cysteine residue on our G3P dehydrogenase. So the cysteine residue is negatively charged. It's going to attack our carbonyl on the G3P. And that's what I'm going to call it. And it's going to attack and it's going to form this tetrahedral intermediate. Again, it's very similar to what you've seen before. The tetrahedral intermediate collapses, and when it does, we're going to kick off our CH bond. Now you would think, oh, why not the CC? Well, the reason why we do CH is because we have that enzyme coenzyme, sorry, right here ready to receive that H. And so now we form this enzyme acyl intermediate, and what we're going to do is we're going to dissociate the NADH and bring in a new NAD plus and inorganic phosphate. So our inorganic phosphate is going to attach itself to this carbon to form that tetrahedral intermediate, and then it'll collapse again, and it'll basically form this free 1,3-BPG, right? All right. So moving on to step 7, we have our 1,3-BPG. We're going to dephosphorylate it. Again, we're going to use this phosphoglycerate kinase. This is the one instance where the enzyme is named for the product where, well, there's multiple instances, but this is the first where we have it phosphoglycerate. So we have phosphoglycerate again and kinase. We're phosphorylating it to form this. But I know it's a substrate. It's just named like that. Do I know why? No, sorry. So the point of this step is that we're removing a phosphate from sugar to produce ATP. So this is step 7. And remember, this is where we're first going to generate our usable substrate level ATP, substrate level phosphorylation to form our ATP. This step is very exergonic because 1,3-BPG is even higher in energy than ATP, and that's because we have this double phosphate repulsion. So it's this phosphate group just wants to leave automatically, so it's going to drive this reaction towards the products. And this is the first time ATP is made in glycolysis, and the resonance stabilizes 3-BPG by distributing the negative charge over 2 atoms, essentially. So it allows this resonance to stabilize it a bit, so that way 1,3-BPG is actually usable and it can form as like a somewhat stable intermediate. But again, it's still energetically much more energetic than the ATP per se. Okay, and then step 8, TLDR, our next step is now we have gotten rid of the 1 phosphate group. We still are left with another one, the 3-phosphoglycerate. Now, what happens? We shift the phosphate to the center carbon. So 3-phosphoglycerate turns into 2-phosphoglycerate. So you could either think of it as a isomerase or, in better terms, a mutase. The difference between an isomerase and a mutase is that the isomerase doesn't transfer a functional group. It just changes the orientation or rearranges the atom, rearranges the bonds in a molecule. Whereas a mutase transfers a functional group. And yes, it breaks the bonds and the functional group into a different part of the molecule. And so phosphoglycerate, again, it's a phosphorylated glycerate molecule, and the mutase, which we're mutating, and not in the sense of like DNA point mutations, but we're transferring this phosphate group into carbon number two. And in the mechanism, we form 2,3-BPG as an intermediate. And this is what's, remember that, no, I'm not going to mention that. I'm going to skip over this, but the mechanism forms 2,3-BPG as an intermediate. And if you remember from hemoglobin, you know what this is used for. And so moving the phosphate. So this is an endergonic reaction where this mutase group, it, it's energetically non-spontaneous, but the reason why it proceeds in the forward direction is because we're removing this right away. So you can think of it as like, again, we had that commitment step in step 4, and now we just want to proceed through it really fast. So we're removing the products for each step super fast that it just drives the reaction forward once you commit to this process. And I wanted, and again, to point out from this mechanism, we form a 2,3-bisphosphoglycerate as a mechanism intermediate, and this could be used as an allosteric regulator of hemoglobin. But the mechanism is a phosphohistidine donating a phosphate to carbon number 2, the hydroxyl group. And then from there, it takes the phosphate group from the other carbon. But yeah. And then step 9, TLDR. So what ends up happening in this is we have 2-phosphoglycerate, which is what we form from that mutase, to convert into phosphoenol pyruvate. Now, what happens is we're losing an OH and creating a double bond to create a direct precursor to pyruvate. So we're basically doing a condensation reaction where we're getting rid of an OH and an H and forming that double bond right here. So enolase is the enzyme that catalyzes the reaction, and we remove water at the end of it. And we form this thing called phosphoenol pyruvate, and it's just called PEP. So formation of PEP is where we remove water to form an enol right here, and it's an endergonic reaction that's reversible, but it's coupled to a highly exergonic step 10, so it's pulled forward. So in other words, this is at such low concentrations because it's being pulled over into the next reaction. And this also, another thing to point out is phosphoenol pyruvate is also a very high energy molecule too, as well. So this is not as much as 3 BPG, but 1,3-BPG, but it is pretty up there as well. And then for step 10, the final step in glycolysis, and then I'll be done yapping. Well, not quite. Our PEP is going to be converted into pyruvate through pyruvate kinase. Again, another instance of where the enzyme is named for the product, not for the substrate. So pyruvate kinase, we take pyruvate, we add a phosphorylation group, hence kinase, and we form our PEP. Phosphoenol pyruvate. And so this is an extremely endergonic step because PEP is an incredibly exergonic molecule with higher delta G than ATP hydrolysis. And so we're going to form our last pair of ATPs, because remember, there's two. We're going to form our last set of ATPs, and we're going to form our pyruvates, which this can be used for the Krebs cycle, but we'll talk about that in the next section. So for step 10, our second substrate level phosphorylation, talking about it more in depth, is we form this ATP at a substrate level phosphorylation, and this is a very exergonic step because it's basically essentially irreversible, where PEP is much higher in energy than ATP. So to go from phosphoenol pyruvate to ATP is super energetically favorable. So it's hard for it to go back the other direction. And here's some info about the energetics. You don't need to necessarily memorize the numbers. You just need to know that PEP is much more energetic than ATP itself. And so it has high energy repulsion due to the negative charges, and pyruvate can tautomerize to form a more stable keto form, further driving the reaction forward. Again, we're reducing the number of products and concentration wise, and this just drives, sorry, we're forming a low concentration of products, and so this just drives the reaction even more forward. And so here's like a review. I would recommend to go through these slides like on your own through like a presentation like this, and just try to see if you know all the reactants, the products, the enzyme, exergonic, endergonic, aim and purpose, like what the whole point of that is. And then here's the practice problem as well. And has the answers on the footnotes as well. So just stuff to look at on your own time, because I know this is already extremely long. I'm trying to condense it, but at the same time, I want to make sure I discuss everything thoroughly. And then the very like auxiliary step, like offshoot of glycolysis that also occurs in the cytoplasm is fermentation. And this occurs in anaerobic respiration, meaning if there's no oxygen, Krebs cycle won't occur, ETC doesn't occur, so we need another way to form our energy. Now, the reason why we have fermentation is because, as indicated in these red boxes, is the formation of NAD+. And so there are two types of fermentation. One is in yeast bacteria, which is this alcohol fermentation, and another one is in like human, animal, mammalian stuff, which is lactic acid fermentation. So for lactic acid, we'll talk about this. It's this top one right here. We're taking our pyruvate and we're making lactate, which is just lactic acid in other words. It's just the deprotonated form. And what we end up doing is we end up using up the NADH to form our NAD plus. Now you may think, well, why are we using our electron coenzymes, right? Why are we using NADH? Why don't we just keep creating more NADHs? Well, NADH is used for ETC. It is not used for glycolysis or Krebs cycle. So there's no purpose of it for just the glycolysis aspect, right? So in order to keep glycolysis running, remember, we need NAD plus for that sixth step, right? With the glyceraldehyde 3 phosphate into 1,3 BPG. You need it for that step, the NAD plus. And if we constantly just drive glycolysis forward without replenishing that NAD plus, we cannot generate any more substrate level phosphorylation ATP. Does that make sense? So if I go back to this general diagram, right? Let me just go really quick to it. So if I go back to this diagram right here, right, we see that we're forming this NADH, or this step is necessary to proceed to the rest of glycolysis, right, to form our two usable ATPs, or net two ATPs that we can use in the cell, right? Well, here's the thing now. If we keep going glycolysis, if we let glycolysis keep proceeding forward, constantly going to the products, we're not going to regenerate our NAD plus. NAD plus is generated usually through ETC, through using those coenzymes to stimulate oxidative phosphorylation. Now, if we don't have those enzymes, if we aren't using those pathways, how can we regenerate NAD plus? NAD plus, and that's through the fermentation. Let me go to this thing. Uh, Give it a sec. Okay. And so how do we generate our NAD plus? That's through fermentation, through either lactic acid fermentation or alcohol fermentation. So now on the other hand, for alcohol fermentation, this is in yeast bacteria. It's a two-step mechanism where we convert pyruvate into acetaldehyde through pyruvate decarboxylase. And you should know the cofactors. I haven't even mentioned, but for like magnesium, potassium, we've seen a couple of those in these previous enzymes. You should know those as well. And same with TPP. We'll explain what TPP is in further pathways, but just know TPP is a cofactor that's commonly found with decarboxylase enzymes. So you can write that down. Anytime you see a decarboxylase enzyme, it will typically have a TPP coenzyme in it. And you can look at the structure of it and all that. So we convert the pyruvate into acetaldehyde, and this is now a 2-carbon molecule. This is not a 3-carbon anymore like pyruvate is. And from acetaldehyde, we're going to reduce it, not oxidize it, reduce it into ethanol to basically turn an aldehyde into an alcohol. And so we use, again, NADH to form our NAD plus. And so fermentation is just the regeneration of NAD plus when ETC, Krebs cycle, is not occurring on its own, when we're in anaerobic conditions. And so the fate of glucose, there's multiple fates, but so far what we've talked about is oxidation via glycolysis into pyruvate. And now what we're going to talk about is ribose 5-phosphate via oxidation using the pentose phosphate pathway. And this is just creating NADPH. So I just want to keep a brief overview before we go into like the very specifics. This is not about memorizing every little intermediate. This is memorizing the bigger picture ideas, right? So there's two phases for pentose phosphate pathway. There's the oxidative phase and there's the non-oxidative phase. The oxidative, look at the name, oxidative, meaning that we're going to be doing a lot of redox reactions, right? So what we're forming here, don't worry about these intermediates, worry about the NADPH. The NADPH, it is distinct from NADH. It is very distinct. Now, NADPH allows us to build biomolecules, whereas NADH is an energy molecule for ETC. That's the distinction. NADPH will help you build fatty acids. It'll be precursors for sterols, for your steroids and steroid hormones. So NADPH builds molecules. NADH is used for ETC, for ATP production via oxidative phosphorylation. So that's the oxidative phase. And there's 3 steps where you end up making, and this is what you do have to memorize, ribulose 5-phosphate. And you form the NADPHs along the way. You don't need to memorize this. I mean, MCAT you should, because they do test you on it. But in terms of for this class, like memorize ribulose 5-phosphate, I mean, it's just kind of good. It's kind of good if you already know this one as well, the intermediate. But for one glucose 6-phosphate molecule, Oh yeah, and that's also another thing I forgot to mention. We're starting at glucose 6 phosphate. So we just did the first step of glycolysis, right? With hexokinase converting glucose into glucose 6 phosphate. Glucose 6 phosphate can now go through this alternative pathway right here that we see. Or it can continue glycolysis. It can choose either or. Or it can choose glycogenesis. So we won't talk about that right now, but just know that glucose 6 phosphate is really this like adaptable molecule. And so in this case, glucose 6 phosphate is going to get oxidized, reduced, all that good stuff. It's going to form ribulose 5 phosphate. And basically, from there, it can either be converted into ribose 5 phosphate, which is used for DNA synthesis, or it can undergo the non oxidative phase to form glucose 6 phosphate. Non oxidative phase is the second part of this, where we have a series of steps that recycles ribose 5 phosphate into glucose 6 phosphate. And it basically interconverts the pentose phosphates and glycolytic intermediates. So we use transketolases, transaldolases, enzymes that just basically change the structure of this ribulose 5-phosphate. So it goes from ribulose to like, I don't know, something else, xylose. I'm just making this up, but it just converts the structure. It doesn't add any phosphates or nothing like that. It doesn't oxidate. It doesn't do any of that. It just rearranges it and it's going to convert it back to glucose 6 phosphate. And so here's like an example. You have ribulose 5-phosphate. This is the non-oxidative, right? Transferring carbon atoms in the non-oxidative phase. So we're taking ribose 5-phosphate, cetoheptulose, fructose. You don't need to know all these. Just know that you're basically adding carbons, taking away carbons to ultimately get to glucose 6-phosphate again. That's all you realistically need to know from this. And it does so through these transketolase, transaldolase enzymes. That's it. You don't need to know. They're not going to ask you and blank each thing out and have you memorize it. No, that's not the point of this. And then these are feeder pathways. So I just want to talk about this. What if we're not working with glucose now, right? Okay. So this right here is talking about what if we don't have glucose, right? If we don't have glucose and we're using some other sugar molecule like galactose, mannose, fructose, there's different entry points into how it gets interconverted so that it can go through glycolysis. And that's all that this is hinting at. The one that you need to know is glucose, obviously, but this is just pointing out like galactose goes through its own pathway to form glucose 6 phosphate. Our mannose it gets converted into fructose 6 phosphate. Fructose gets converted into glyceraldehyde dihydroxyacetone phosphate to form our G3P. So all these molecules will enter the glycolysis path. So you can think of like glycolysis as like the pillar, and then the other stuff are branches that lead into it, right? So that's what the basic gist. So that was glycolysis. I know it was a lot, but if you have any questions, like please come to my office hours. And I know I seem very segmented, but that's just because there's so much information to present. Just post any questions on ed or whatnot, and just rehash the video as well if you need to go back on like some smaller steps or whatever. All right, change the scenery for a bit. Lecture 9, citric acid cycle. I feel a lot more organized for this, but let's move on into it. So we just talked about glycolysis and how we split the sugars. We had partial oxidation, and we're now basically at pyruvate. And what we're going to have is, I know it's called pyruvic acid, same thing, synonymous. Now, this is in the cytoplasm. This is important, what I mentioned before, that the pyruvate has to go from the cytoplasm into the mitochondria where all the Krebs cycle enzymes are at. So that's what we're going to start talking about. So this is the second step in cellular respiration. And so just giving a brief overview, the mitochondria, it is the powerhouse of the cell, obviously. If you guys didn't know. Basically, it's the site of most energy yielding oxidative reactions. So ETC is in here, Krebs cycle, where we generate the most coenzymes NADH, FADH2, and the mitochondria itself is double membrane. Basically has three spaces. We have the extracellular matrix, which is outside the mitochondria. We have the intermembrane space or intermembrane space, whatever you call it, and then we have the mitochondrial matrix. So there's three like spaces, I guess you could say, with this double layer. Glycolysis occurs in the cytoplasm, and again, as I mentioned before, pyruvate has to get inside the mitochondrion. And the blah 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 blah. The citric acid, yes, TCA occurs inside the mitochondria. This is an important thing to note: is that the inner membrane is impermeable, meaning that pyruvate actively is transported from the cytosol into the mitochondrial matrix. So there is a symporter that transports pyruvate and a free proton from you can even think of it as the inner membrane space. So this space right here that's outside this like inner part, this space, there's transporters between these spaces where they take the pyruvate, the H pluses, and push it inside as a symporter. Now, this is important to note because we know that we're building the proton concentration gradient. Not to get too far ahead, but we're building this proton concentration gradient on the intermembrane space. So just something to keep in mind that the symporter is almost acting against that proton motive force that you're developing in ETC. So something to keep in mind. And then, so that's that. So prepping for the TCA cycle. So let's say we transported pyruvate into the mitochondria, and now the pyruvate's going to come across this very complex enzyme called pyruvate dehydrogenase complex. And this complex forms three enzymes that are put together. And I don't know if I have an image here. Unfortunately, I do not. But there's images online where you can see this like this giant enzyme. I think it's on the lecture 9 like homepage. Like before you click the lecture slides, I think it's that like image that shows there if they kept the same images. But I digress. So there are three enzyme names you definitely should know. E1 is pyruvate dehydrogenase, so you're gonna oxidize the pyruvate. Second thing is dihydrolipoyl transacetylase, meaning you're gonna transfer the acetyl group. And then next one is dihydrolipoyl dehydrogenase, where you're gonna now reduce the cofactors. So that's what these three are used for. We'll go much more in depth breaking each down. I'm just giving you the names. You should definitely have them memorized. And there's five cofactors used. There's TPP, and remember how I mentioned in glycolysis, TPP is normally used for decarboxylation enzymes. Well, in this case right here, where it's highlighted, this orange box, this is a carbon dioxide group. Carbon dioxide is decarboxylated, so TPP is going to be used for that action. Then we have CoA, which is essentially a reactive thiol group forming a thioester to our pyruvate, the other two carbons that are on this. So as you can see right here, this forms a thiol group. And this is important because thiols are somewhat energetically active, not as active as ATP, but it provides enough energy to go through the rest of the Krebs cycle. This will become important in a little bit. And then there's lipoic acid, which this is where the name dihydrolipoyl comes in part. And as Solomon likes to say, it's like an arm that can rotate essentially, but it allows for basically this pyruvate dehydrogenase complex to have a sense of like circuitry in a way. Like it goes from here to here to here to here. That's all that this lipoic acid does, and it allows for us to utilize the thioester and to attach this CoA enzyme into it. So these two work in tandem to form our acetyl CoA that's going to be used for TCA. And then the next thing is going to be DH2 and NADH are going to be produced from this. So now talking on to the cofactors, this is where I'm going to break each one down. So TPP is this one on the top right, and it has the thing that the only thing they will have you do is recognize it, not draw it out. They will never ever ask you to draw this TPP out, not in MCAT, not in this, just for you to recognize it. And the distinction of the TPP is this thiazolium ring, which is essentially almost like a, not necessarily thioester, but there's a sulfur. It almost resembles like a histidine, right? The histidine ring. So you can think of it like that, and then it just has a sulfur inside of it. That's how you recognize a TPP molecule. And it has the phosphate groups attached here. It has almost like a, I don't know, an aromatic ring with nitrogens inside of it. That's what I'm getting from it. But the important group is this thiazolium ring, which allows you to do that decarboxylation action. And then this is coenzyme A, which is going to be attached to acetyl aldehyde or acetyl CoA. That's why we have the CoA in there in the first place. And again, this is the full enzyme. You just need to recognize it. That's about it. Moving on to coenzyme A. So the only thing, you just need to recognize it and just know that it has a pantothenic acid in the middle. So this group is very important because it allows us to attach this beta-mercaptoethylamine, which all it is it allows us to form this diol group that is going to attach itself to acetyl CoA. So if we were to just say everything to the right of this SH, right, is just going to turn into CoA, then you can see how it's attached to this acetyl CoA molecule, right? The H, we're going to get rid of this H, and instead of what's going to replace it is going to be our acetyl group from the pyruvate. So as you can see here, this is from our pyruvate that's been decarboxylated first, and now it's being attached to the CoA enzyme. So this is what it is. But this CoA would be this whole representation here. And as you can see, it's in, what's it called? Adenosine. It's a nucleoside being attached to it. And as a matter of fact, nucleotide right here, because it has the phosphate group. So it's a nucleotide plus pantothenic acid, and then this beta-mercaptoethylamine that allows the CoA, that port to the right to be attached to our acetyl group. And then the last one is lipoic acid. So it's this thing right here, this black part, and it's essentially attached to a lysine residue. So it's almost like forced on. And this is part of just this enzyme 2 complex. And like Solomon was saying, it's just like a stick or whatever that goes around in circles. But at the end of the stick is where I don't like using that terminology, but anyways, or lever. But at the end of it, there's this sulfur groups that are able to get reduced. So right now it's in its oxidized form, and it could get reduced. And when it's reduced, we can acetylate this and be able to almost extract the electrons and to attach this CoA enzyme because again, it's also this part of the group. So this will all make sense once I start breaking it down later on. So as you can see, this is an overview of the pyruvate dehydrogenase complex, like in a cartoon form. So, yeah, I see. So I'm not gonna go and we'll keep an overview. Let's do that. I think that's the best way to tackle this. So we have our pyruvate right here, right to the left. And what we're going to do is first decarboxylate it. As I mentioned, we're going to be using TPP to achieve this. And then we're going to release our, what's it called? Acetaldehyde. That's why am I forgetting the name? Acetaldehyde is going to be released from our first enzyme. So remember, this is the complex. So E1, the E1 is the pyruvate dehydrogenase part of it. And it's going to basically, again, reduce pyruvate because as you can see, we have 2 electrons being owned to carbon, but then it turns into 3. So it's technically being reduced. And then from there, what's going to end up happening is we're going to oxidate this acetaldehyde by attaching it to that lipoic acid from E2. E2 contains our acyl lipolysine, or we're going to form this acyl lipolysine because of this oxidized lipolysine that's going to attach itself to the acetaldehyde. So here's where we are right now. And as you can see, it's oxidized because that carbon now only owns 1. The sulfur, again, is more electronegative than carbon, and it's going to take those electrons away from it. So now we have this arm going around, and as it's going around, we see that the acetaldehyde is going to exchange with coenzyme A. Remember, this is a reactive thiol group, so it's going to basically swap places, and the lipoic acid is going to get reduced again. This lipolysine is going to get reduced, and now we have 2 electrons here, basically. And what's going to end up happening is we're going to transfer those to FAD. And FAD is part of this E3 enzyme, the dihydrolipoyl dehydrogenase E3. And the FAD is going to pick up those 2 H's, or the electrons essentially. They're going to oxidize our lipolysine and turn it back into this reduced or oxidized form. And they're going to take those electrons and then take them through the E3 enzyme and pass them now to NADH. Remember that we said that NADH is freely able to travel throughout the cytoplasm. It's stable enough, whereas FAD is restricted. It cannot like translocate to different areas. It can't do that on its own. So that's why we have this last step, or else we would have just finished with these 2 enzymes and we wouldn't need the third. That's the whole reason why we need this third part. And yeah, so in this whole step, we're seeing that we're performing another redox reaction, essentially, where we're changing the central carbon atom right here, carbon number 2, from 2 owned electrons to now 1. And that's because we're attaching, again, this thioester. The sulfur takes all the electrons here. The oxygen obviously takes all of them. And then it's equal sharing between these CC. So it's just 1 right here that it owns. So that's like an overview. Now let's go into detail of everything, like just breaking it down step by step. So first step is PDH E1. So E1 again is that pyruvate dehydrogenase. And what we're going to do is we're going to take our pyruvate, it's 3 carbons, we're going to decarboxylate it, meaning we take 1 carbon off to form that carbon dioxide. And we're going to get basically like this, it's connected to the TPP. It's called a hydroxyethyl TPP. You don't really need to know that. You just need to know that you're forming an acetaldehyde that eventually gets transferred onto our E2 part of the enzyme. And I think that's everything I needed to say for this slide, at least. And our acetaldehyde will be transferred onto this oxidized lipolysine, reducing it by 1. So right here, you see that they'll be reduced here, but it's still oxidized here, technically. So we have now this E2, which is transacetylation, meaning that lipoic acid will transfer acetyl, the 2 carbons, to free coenzyme A. So it's this number 3 right here, where we're taking our acetaldehyde that's attached to this acetyl lipolysine, and what we're going to do is we're going to essentially swap this S for this one on this acetaldehyde. So this is where we form, instead of acetaldehyde, we form acetyl-CoA. And this is what we're going to use for Krebs cycle. So this is like the first step now where we can let this molecule go freely into the mitochondrial matrix and do the Krebs cycle. And recall that this H that was on the that free coenzyme A, it's going to be transferred onto the lipolysine, and the lipolysine is now going to be reduced. So now we have this very high energy, not necessarily like high energy as an ATP, but just where there's a higher electron density, and it's going to be transferred to E3, which is this next one, the dihydrolipoyl dehydrogenase. So essentially, we're taking our dihydrolipoyl, our two lipoic acid arms, whatever at the ends, the SHs. We're going to take those H's. We're going to take the energy and transfer them onto FAD to form FADH2. So now from this, we're going to get the we're going to get the oxidized lipolysine right here. So now it's going to form this cysteine or the double disulfide bond, and this is the oxidized form, and those H's are going to get transferred to FADH2. As I mentioned in the overview, these are localized. These cannot travel freely throughout the mitochondrial matrix. So what we have to do is do this final step of transferring the FADH2s onto NAD+, and this is going to form our NADHs that we need, and that's where we get the +2 in the overall reaction. Oh, this is the image that I wanted to show you of E1, E2, E3. But yeah, so this will allow us to use these coenzymes for ETC. I think that's everything for that. Yeah. So this is again, pyruvate dehydrogenase complex is a multi-enzyme complex that allows us for this first step of Krebs cycle. This is just to get in the cycle. So like there's like one step that allows us to like commit in essentially. And so this multi-enzyme complex provides rate enhancement because it's short diffusion distances. As you can tell, everything is super close together, so it's very systemized and everything. And it's coordinately controlled, so everything happens for a reason, structurally, mechanistically, all that. And it's inhibited by NADH, acetyl-CoA, and fatty acids. Now, why? NADH is a high energy molecule essentially. You can think of it like that. And acetyl-CoA just is a product inhibition classic. But if we have high energy and we have a lot of product, then we don't need to create more. You know what I mean? So in this case, if we have a lot of acetyl-CoA or if we have a lot of NADH, we don't need to go through this Krebs cycle as much. We can slow down, just let the cell consume the extra energy that we have. And then fatty acids, you'll learn about this in beta oxidation. I don't want to confuse you with extra terminology, but fatty acids will get broken down into acetyl-CoA via another process. So if there's a lot of fatty acids, then there's no reason to go through this glucose pathway. We can just have those free fatty acids do their thing. And then it's stimulated by NAD plus and free CoA, which are signs of low energy. So that's the way you can rationalize a lot of these regulations, is that we're talking about cellular respiration. We're talking about breaking down macromolecules into usable forms of energy. If we have high energy, we don't need to break down those macromolecules. As a matter of fact, we would probably build those up into like glycogen stores, into fatty stores, cholesterol. We would use those for anabolic reactions if we had a scenario where there was high energy. And then PDH kinase and phosphatase. So these are two proteins, two separate ones. PDH kinase will be activated by ATP, NADH, and acetyl-CoA. And it inactivates our E1, the outer portion of this complex, by phosphorylating it. And then phosphatase is the opposite of the kinase, meaning it'll remove that phosphorylation. It'll be activated with signs of low molecules, so like AMP, ADP, NAD+. So both are part of the enzymatic complex, and these will allow for either phosphorylation as an inhibition or dephosphorylation, which is activation. So just know that these are the mechanisms by the way these regulators can regulate it, if that makes sense. So these regulators, ATP, NADH, AMP, ADP, NAD+, they don't just bind onto the enzyme itself. They have their effects because of these two proteins. So PDH kinase and phosphatase will exert their effects, and these are respective based on which molecules are more present. And here is the citric acid cycle. I know it looks very complex, but we're going to break down, and I'm going to give you like the easiest mnemonic that I yet to this day, like whenever I haven't reviewed it, whenever I don't review the citric acid cycle for a long time, I just remember the mnemonic and then all the enzymes come back, all the substrates, so I'll help you. And then a helpful tip is to count your carbons on your reactants, intermediates, and products. That way you understand where they're coming from, where they're leaving. Was it from a previous cycle? Was it from the same? And I'll break that all down in a little bit. So this is the useful mnemonic for intermediates. And trust me, this is a good mnemonic. Citrate is Krebs starting substrate for making oxaloacetate. I don't know if I counted that right. I was doing random finger motions. But essentially, you're starting with citrate, and then is Krebs, so isocitrate, alpha ketoglutarate, starting substrate, succinyl-CoA, succinate. So you have to remember the order. That's like the one thing you have to keep in mind. 4, fumarate, malate, oxaloacetate. And then, yeah, you end off. So you end off with like the starting and the end. You know that the first part is citrate, and then the last part is oxaloacetate. But this is a phenomenal mnemonic that I just highly recommend you guys remember. So we're going to go through each step. The first one is just keeping it same format, TLDR, and then the breakdown, greater breakdown, so you can reference it on your own time. So this first step, remember those acetyl-CoAs we formed from the PDH complex from turning pyruvate into acetyl-CoA? Well, now these molecules can interact with oxaloacetate in the mitochondrial matrix. Remember, we are in the mitochondrial matrix. We are not in the cytoplasm anymore. So these two will combine via citrate synthase. So it's kind of hints at the name. It talks about the product. So it'll form citrate from the combination of acetyl-CoA and oxaloacetate. Now, this process is exergonic, meaning it's very spontaneous. It'll happen on its own. But why is that? The reason why is because, again, remember this thioester bond? It has a lot of energy, not as much as ATP per se, but it has enough energy that it wants to be hydrolyzed essentially, that it can be, and basically it'll attach to the oxaloacetate. And another big thing is that we're combining 2 carbons and a 4 carbon, so 1, 2, and then 1, 2, 3, 4, to form a 6 carbon molecule. 1, 2, 3, 4, 5, 6. And we're adding water to hydrolyze this thioester bond, and it'll release our coenzyme A, and it'll reduce it essentially. One important thing I want to note for like the details is that this reaction is irreversible, meaning it does not occur in the backwards direction. Once you hydrolyze the thioester bond, it's very hard to, it's almost impossible to put it back on. And so this step is going to be obviously highly regulated as well, and it's stimulated by, again, low energy molecules, ADP, and it's inhibited by high energy molecules, NADH, ATP. Again, if there's a lot of product, it's going to inhibit it through feedback inhibition, product inhibition. So citrate will inhibit it, and it's also inhibited. This is a special case, and you should memorize this. I wish I could tell you why, but succinyl-CoA, a downstream product, will also inhibit citrate synthase. And I think this has to do with muscle now that I'm mentioning it, but I don't want to like conjecture too far. And so in general, high energy will inhibit the citrate cycle, low energy will activate the citrate cycle. And then moving on to step 2, we now have our 6 carbon molecule that's been combined. And what's going to end up happening is we're going to see a shift. Citrate is. So we're going to see a shift from citrate to isocitrate. So how is this achieved? Through an enzyme called aconitase. And aconitase utilizes the water elimination and hydration reactions to basically isomerize our citrate molecule into isocitrate. So dehydrate, then rehydrate, remove the water, then add the water. That's basically this mechanism. And this aconitase, the way it works is it contains iron sulfur clusters to coordinate this entire reaction. So there's a reason why iron sulfur clusters are important, and I'll explain why. So there's a concept called prochirality. This is a little bit different. It's very similar, but not quite the same. So prochirality just means that we have a molecule right here, citrate, is achiral, but it gets converted into a chiral molecule via isomerization. So what that means is right now, if we were to look at this carbon, right, this is the achiral carbon that we're talking about. It has an OH, it has a COO, so already 2 different groups. Then you think, oh, CH2CO minus, that's one group. Oh, this one looks different, but it's not. It's just the notation. It's messed up, but that's how they're trying to like just to show you why these OHs and H switch. But this is achiral. These 2 groups, the top and the bottom, are the same. So if they're the same, we know it's a meso compound, so it's achiral. But if we swap the OH and the H, we see that now it's chiral. If we look at the center carbon atom, this is a carboxylic acid, this is a methyl carboxylic acid, this is an H, and then this is the carbon, but it's not a methyl or ethyl, whatever it's called. It's an H and then an OH. So now these are 4 different groups. And so because there's a chiral product, that means the enzyme, in order to accommodate for that, has to account for that asymmetry. So essentially, only one product is formed, and the carbon substituents have to bind to the enzyme in a specific pattern. So there is spatial orientation to this, that the enzyme itself is altered at the active site. So the active site, it will force the citrate to kind of fall in a specific direction to allow for isocitrate production. Hopefully that makes sense. So step three, TLDR. So now that we took our citrate, converted to isocitrate right here, and now we form this prochiral molecule, or chiral in a sense. This is chiral. We're going to take the 6 carbons and we're going to decarboxylate it. So we're going to go from 6 carbons to a 5 carbon alpha ketoglutarate plus carbon dioxide. And so the carbon dioxide is going to be our decarboxylation portion of the aspect. But on top of this, we're going to also reduce NAD to NADH. And so we're going to form our first coenzymes of the actual loop in the Krebs cycle. Remember, PDH also creates its own as well, but that's not part of the loop. This is the first one as well. So energy is now stored in NADH. Yeah, I will go through that. So for this one, this isocitrate dehydrogenase is is utilizing that isocitrate, the OH, the hydroxyl that we switched and isomerized, and it's going to basically oxidize this, and it's going to allow and prompt the molecule and the enzyme to decarboxylate it from there using TPP and manganese. And so we're going to form again that 6 carbon isocitrate is going to be converted into a 5 carbon alpha ketoglutarate. And I just want to highlight this process is extremely exergonic, so it's going to be essentially irreversible. And it's inhibited by ATP, but it is activated by calcium. This is from skeletal muscle when you apply this, but I won't go into depth. That's the only reason why calcium would activate it. And then secondly, ADP, AMP. Actually, I do know why, now that I'm thinking about it. So calcium activates it because whenever we have a surge in our sarcolemma, the cytoplasm of these muscle cells, when we have a surge, we want cross bridges to happen. That means we're burning through ATP because if you remember tropomyosin, actin, troponin, all that stuff, the cross bridges are going to start happening when there's influx of calcium. So when we have a huge surge of calcium, we want more ATP to be produced, right? So we need to rely on Krebs cycle in order for that ATP to occur. So this is why calcium would activate isocitrate dehydrogenase. Do I know why it doesn't? I don't know why that wouldn't activate the other upstream enzymes like citrate, synthase, or PDH, but just know that for this case right here, that's why it would activate isocitrate dehydrogenase. And then also low energy molecules like ADP, AMP would also activate it too. And then step 4, this is a TLDR. We took our alpha ketoglutarate, we're going to turn it into succinyl-CoA. This is the last part where we see decarboxylation within the loop of the Krebs cycle. So citrate is Krebs starting. So this is where you have to memorize that it's just succinyl-CoA, not succinate. And again, we're taking the 5 carbon molecule, converting it to a 4 carbon succinyl-CoA, and our carbon dioxide, this is where we see another release of carbon dioxide. And we're going to oxidize the sugar again, but we're also going to now get new reduced coenzymes, NADH, as well. And we're going to replace that carbon that we lost with the coenzyme A. This is where we introduce it again. And now you may be thinking, well, wait, this is very, very similar. Using CoA, NADH, all that stuff. It's very similar to PDH. Well, that's exactly what I was about to say on the next slide, is that alpha ketoglutarate dehydrogenase complex is using the identical mechanism to PDH. So you should already know this, and I won't go into the details of it, but you should know why each step occurs, why we have decarboxylation, the lipoic acid reduction oxidation, and our reduction of cofactors as well, using FAD first and then to NADH. And then we also put the coenzyme A onto the alpha ketoglutarate, or the backbone of it, the 4 carbons to form succinyl-CoA. So this reaction is very exergonic. Again, irreversible, essentially keeps going through. And removal of CO2 is the highly exergonic portion that pushes it forward, and it produces NADH. The regulation is, again, inhibited by succinyl-CoA and NADH, so high energy molecules, either product feedback inhibition or NADH, which is high energy. And then it's activated by calcium, again, with that rationalization that if our muscles are contracting, we need to use a lot of ATP. And the only way they can contract a lot is if we have that calcium influx. So if we have a huge calcium influx, it's going to bind onto these receptors and enzymes like alpha ketoglutarate dehydrogenase complex and isocitrate dehydrogenase to stimulate this pathway. These are highly exergonic but tightly regulated too. So that's why calcium would almost counteract and activate it. And then step five, TLDR, is we're taking our succinyl-CoA and we're forming succinate. We're getting rid of our CoA that we have in here, the thioester, and that bond is going to allow us to power GDP plus PI, like condensation, to form it together to form this GTP. And GTP later on will form ATP. It's just like a loop, an exchange, just in the similar way that FAD to FADH2 will exchange the electrons that it now gained with NAD plus to form NADH at the end. It's like an quick exchange like that with this part right here. But please do remember that succinyl-CoA synthetase will form GTP, not ATP right away. In the end, the net result is ATP, but for this, just know it's GTP. The reason why we use synthetase instead of synthase is because synthetase is used for anything that generates GTP or ATP, whereas synthase only generates specific molecules without producing that ATP or GTP directly. So reaction, it's essentially you cleave the thioester, which has a lot of energy, and you use that energy to phosphorylate a GDP into form a GTP. And this is exergonic due to that thioester cleavage. And this is, again, another example of substrate level phosphorylation. And yeah, here's an example, like side note of synthetase versus synthase. And yeah, just rehash that. But again, it's exergonic, but it's also reversible too. But the reason what drives it forward is we also get rid of the succinate right away. So this is the three-step mechanism to oxidize an alkyl chain. So they're essentially like grouped together, the next three steps. So we have succinate, citrate is Krebs starting substrate. So step number five, or step number six, sorry, after five is done. So starting substrate. So this is succinate. Four, fumarate, making malate. So this is going to be our fumarate, it's going to be our malate, and then oxaloacetate. So again, that mnemonic just allows this to be simplified. But the whole premise is that we now have this succinate, right, over here, and all of its single carbon molecules, right? Well, how can we harness more energy from this or turn it into oxaloacetate? Well, we use the classic dehydrogenation, sorry. We get rid of one hydrogen from each carbon to form this double bond, as you remember from Orgo 1. And because we're getting rid of one hydrogen, we can transfer it onto our FAD coenzyme. And then from there, we can add a water to hydrate it again to form this hydroxyl group, and then oxidize it to form NADH, or its last energy carriers. So we'll go through each one in depth. That was just an overview. So step six, we have succinate right here. What ends up happening is, again, we remove H's and form a trans double bond to create FADH2. And it takes our four carbon and it stays four carbon. So remember, oxaloacetate is four. So we're no longer dealing with decarboxylation. We're no longer dealing with TPP. We're just trying to harness the rest of the energy possible. So for redox, we're basically oxidizing the carbons, reducing the FAD, and that's where the energetics are going to be stored and passed on to the ETC. And I will point out, succinate dehydrogenase is complex number 2 in ETC. So this is coupled to not necessarily proton transfer, not that, but it's coupled to the ETC on the inner membrane of the mitochondria. So good side note to have. And again, it's reversible. It's delta G naught is 0 kilojoules per mole, so it's a high reversible reaction, but it drives forward just because we're getting rid of the products. Then the next, so this is the more in detail. One key fact, malonate is a competitive inhibitor of this reaction. So it can't lose two hydrogens and cannot be oxidized to that double bond just because of the way it's like stable, the reactivity or whatever. Just know that this competitively competes with succinate for this active site and succinate dehydrogenase, and it prevents this reaction from occurring. So that's just a little side note. But moving on to that, when we have formed fumarate, I know I skipped over what I was going to say is only trans product is formed. This is important. So one thing I will say, actually, I don't know why I didn't touch up on this. Succinate will only form the trans product of fumarate. And this is important because that trans product of fumarate will form a specific type of malate. That's the only reason why. It doesn't form a cis fumarate. It only forms a trans. So this is where it comes into importance, is that that trans fumarate in step 7 is going to become hydrated, but it's going to form our L malate, not D malate, L. And that's very important to know, is because it could be like an easy multiple choice. Know that it will only form an L malate. And this is where we break that double bond by hydration. So we add the OH, then the H plus separately. So it's not necessarily like a water, it's like a separated molecule. Or no, it is a water molecule. I don't know why it's OH. But anyways, we're adding water molecule to this if we were to take just the overall reaction. And we're forming this carbanion transition state where it's forming like this like tetrahedral structure that's like very volatile. This carbanion right here, and we add the proton to basically protonate it, but it allows for the formation of L-malate. And it's highly stereospecific enzyme, meaning the substrate is only transfumarate and the product is only L-malate, not D-malate. And it's an exergonic reaction, pushing it forward and dragging the reactions before it towards the products. And then the last step, which is forming oxaloacetate, we take our L-malate and we're going to use L-malate dehydrogenase, not a complex enzyme, and we're going to basically reduce our NAD plus into NADH, and this will oxidize our malate to form oxaloacetate. So that's basically this. Yeah, and then it's rinse and repeat. Oxaloacetate feeds into step one, which is citrate synthase. You combine acetyl-CoA, oxaloacetate to form citrate. And again, a lot of these enzymes are named for their reactants. So that's the easiest way to memorize it as well. And then this is in depth. This enzyme or this reaction is extremely endergonic, which means that it doesn't want to go forward. But the only reason why is because we remove oxaloacetate when it combines with acetyl-CoA in the first step. And remember, that one is irreversible, so it's not going backwards. There's no like backlog. You know what I mean? So if once the oxaloacetate combines with the acetyl-CoA, it's going to stay at citrate. It's not going to go back. So we're essentially getting rid of the oxaloacetate. There's none. So malate's being pushed forward, despite it being endergonic. And so these are, yeah, again, this is just a table summarizing the TCA cycle regulations. Again, the calcium has to do with muscle contraction. That's why we see it as at some point at activators. And inhibitors, there are some of them that you have to know, like fatty acids. That will make, I wish like beta oxidation was presented because that would make so much more sense. But just know fatty acids, they break down into acetyl-CoA, and that's what inhibits PDH by feedback inhibition. So that's the only reason why. And then you have to know the inhibitor, the special ones for succinyl-CoA, alpha keto dehydrogenase complex. Just memorize this table for sure. And then TCA intermediates, this is a good side note, are often removed to be used in other biochemical reactions. And when they're removed, they have to be replenished. They have to be fulfilled again. And how is through these anaplerotic reactions, which are designated by red arrows. And they use different enzymes, right? So you may say, well, pyruvate's entering the Krebs cycle as acetyl-CoA. Doesn't that regenerate the products? No, because the two carbons from acetyl-CoA are eventually going to be the two carbons that come out from carbon dioxide. Does that make sense? So remember that we always go from 6, from that citrate, to 4, right? And what are the two carbons that we lose? Where do they come from? Those are from the acetyl-CoAs. That's where you're feeding into it, and that's what comes out. It is not any of the carbons that were bound to this oxaloacetate. Does that make sense? So you may say like, oh, can't pyruvate just replenish it via acetyl-CoA? Well, no, because the carbons that are introduced by acetyl-CoA are going to be the exact carbons that leave as carbon dioxide. So always know that in order for carbon dioxide to be generated, it has to come from acetyl-CoA, not from this oxaloacetate. And so the only way we can replenish it is by using pyruvate carboxylase, not decarboxylase. So it doesn't use TPP. Use something else called biotin. We'll talk about that later. And pyruvate is going to you're going to add a carbon to it. Remember, pyruvate's 3 carbons. And you're going to add one, so carboxylating it to form this oxaloacetate. And then we also have PEP that could also be carboxylated. Remember, PEP is a 3-carbon molecule in step 8 or 9, I can't remember. But it's going to be carboxylated to form this oxaloacetate as well. And same with PEP carboxykinase too. These are 2 different enzymes, 2 different pathways that allow us to replenish oxaloacetate. And pyruvate can also be converted to malate via this malic enzyme. And again, these are anaplerotic reactions, the red ones. The blue arrows are not anaplerotic because they take away or they don't actually add themselves to the intermediates. They don't let it build up. Because, like, for example, the acetyl-CoA, 2 carbons that come in must come out. That's always remember that. Moving on to the next one. So anaplerotic reactions. TCA is a cycle. Every molecule that is removed for every other reaction must be replaced. So I already said all that. Let me see. Okay, so biotin. So remember that biotin is always a cofactor that will be used in carboxylation reactions. So it's very structurally similar to our TPP, right? Remember it was that histidine with the S, like this is me vaguely remembering. As you can see, there's the S, and this almost looks like histidine. I don't know. It looks very similar. But biotin allows us to add a CO2 to a molecule, not take one away. It's always something to keep in mind that um, We have carboxylase, carboxykinase, carboxylate. Malic enzyme is a special name. Now I don't know why, but that essentially you're adding a carbon to it. But all these, you're adding carbons, and they're technically anaplerotic reactions because you're taking the three carbons and you're forming the four. Okay, so pyruvate carboxylase. So TLDR. Before I move on to this other cycle that I was about to mention, the goal is to add one carbon via this carboxylase enzyme. So the way it works is we're using biotin, right? Remember how I mentioned it before? That's very like similar, structurally looks the same as TPP or similar, vaguely. And we're gonna use, where do we get our source of carbon for that addition of carbon? That's through bicarbonate. So biotin is gonna allow us to attach the carbon from bicarbonate onto our pyruvate. So essentially, the first step is we're adding a phosphate to bicarb. So right here, you can see these arrows are showing that. So we're phosphorylating our bicarbonate to form a carboxyphosphate right here, as you can see. And this is not, the biotin is not in this picture yet. It's just this is happening on its own. And so carboxyphosphate will basically, you'll form this like intermediate that collapses on itself, and this will form our carbon dioxide right here. Now the carbon dioxide can interact with the enzyme. Right here we see sorry, biotin, that like a nitrogen on it. I think it looks like it's, I know it's not. I know it's not, but it looks like it. And this nitrogen will attack the carbon dioxide, and it'll attach itself to form this intermediate, this acyl intermediate, carboxybiotinyl enzyme. It's just forming an intermediate attached to the enzyme that's covalently attached. After step three, right? That our carboxyl-biotinylated enzyme. What's going to end up happening is we're going to flip over our biotin, and so now the carbon dioxide is on the right side, right on this diagram, right over here. Now, what's going to end up happening is the carbon dioxide is going to leave, and it's going to pass on some of its electron density into the biotin itself. So you can see right here with this arrow pushing, that we now have this. Can't remember the name. It's a like enamine or something like that. I can't remember the name, the formal one. But point is that our negative charge is on the oxygen, right? And it's gonna kick back, and instead of interacting with the carbon dioxide, because if it did, it would just go backwards. Instead of doing that, it's gonna attack and it's going to deprotonate our pyruvate, and it's gonna form this essentially PEP or pyruvate enolate. Sorry, I don't know why I was thinking of that. It's going to form our pyruvate enolate right here. So that's what's going to end up happening after step five, or step six, sorry. And the pyruvate enolate is going to collapse on itself, and it's going to attach itself to the carbon dioxide. And now we have oxaloacetate, and this is released. And again, we regenerated our biotin. All it has to do is just flip back once it leaves, which it probably does based on interactions or whatever. And yeah, that's essentially the mechanism. Hopefully that made some sense. And then moving on to the glyoxylate cycle. So this allows for the conversion, and this is only in invertebrates, so not in vertebrates, so not in humans, none of that, like in plants, I'm thinking. So in glyoxysomes, specific cell types, or organelles, whatever, I don't even know, it allows for the conversion of two carbon molecules to four. So it'll take our acetyl-CoAs from fatty acids right here, right? And what it will do is it'll combine two of them together to form citrate. Or sorry, it'll combine acetyl-CoA with oxaloacetate to form citrate. And then from citrate, it'll go through isocitrate. So uses, again, the same enzymes in Krebs cycle, but we have this isocitrate lyase now. This is a new enzyme where it'll essentially cleave it, and you'll form a succinate that's anaplerotically replenished TCA cycle. And you'll form this glyoxylate molecule as a result, and that stays in this loop. And then you'll have another acetyl-CoA, another one from fatty acids, beta oxidation right here. This will come in and form our malate and form our oxaloacetate, and then the cycle can continuously continue. But this is exactly why humans cannot survive on primarily fat diets, because primarily fat will only break down into acetyl-CoA. There are special kinds of fats that do allow you to generate our succinate, but those are special fats that are not prevalent in the diet that we have. So we can't rely on that to survive. So with fatty acids, we're going to form acetyl-CoAs, but as you remember, TCA, whatever carbons we put in are the carbons that come out as carbon dioxide. So you always have to remember is that the carbon dioxides that come out are from the acetyl-CoAs that we put in. If we don't have any acetyl-CoAs, right, then the reactions just stop. And then there are two unique enzymes to this. And as I mentioned, isocitrate lyase and then malate synthase. It allows us to combine the glyoxylate and acetyl-CoA to form malate. So those are the two that you just need to remember that allow this glyoxylate cycle to occur. And it's two enzymes that we don't have. And this is the net reaction down below. But honestly, like if you just memorize this like cycle and understand it intuitively, this will come to you easily. And that's all for TCA. It's a 50. How long is it? Like two and a half hours. I am so sorry for the delay, but hopefully I was able to make these videos a lot more I don't know, to break it down in another way, hopefully, and hopefully I didn't just confuse more people. If so, I have office hours this Wednesday. Probably be Satellite Mez because booking they didn't keep my booking stuff at Greg, so and spot was taken. So I'll probably do Satellite Mezzanine from 7 to 9 or something like that. Whatever my time is, I can't even remember. But yeah, I'm sorry guys. I will be on top of it for lectures 10 and 11 this week. I know photosynthesis is going to be more on the tougher side just because who cares about photosynthesis. Okay. Anyways, I hope you guys have a good one. See ya.